In this video, we will be taking a protein sequence and comparing it to other proteins with similar sequences. We will then take the sequence of the 5 most similar proteins and perform a multiple sequence alignment using the Clustal Omega program. Using the multiple sequence alignment, we will be able to compare which sequences are most similar to each other and construct a phylogenetic tree depicting the relationship between these proteins. First, go to Uniprot. Type the ID of your chosen protein in the search bar here. I will be using the sequence for the urease subunit alpha protein in Helicobacter pylori. On the left, click on the sequence section in the quick access list here. This will bring you to the section with the sequence. On the right of this section, you can perform a blast run of your sequence directly from Uniprot. Hit go. Then hit run blast. On this page, you will notice the job identifier in the center. This is associated with our current blast run. If at any time in the future we would like to bring up the same results without running the blast search again, we can do so with this ID. This may take a few seconds or a few minutes depending on your sequence. Once the run is complete, you will be greeted with the overview section. Scroll down a little bit to find the alignments section. This section contains the alignments list, which shows the most similar proteins based on their sequences. On the right, you will find the E value, the score, and the percent identity of each of the sequences in relation to your own sequence. Next, we want to download a FASTA file containing the sequences of the top 5 hits. We will use this FASTA file to perform the multiple sequence alignment later on. Select the top 5 hits, like so. Note, do not select your query. Click on the Download button. Ensure that you're only downloading the selected 5, that it's in FASTA format, and that it's uncompressed. Then hit Go. Right-click this page and hit Save As. Change the file extension from txt to FASTA and hit Save. Now that you have downloaded the FASTA file with the sequences, we will compare them to each other by performing a multiple sequence alignment. A multiple sequence alignment is an alignment of 3 or more biological protein or nucleic acid sequences. Usually, these sequences are of similar length, and from the output, evolutionary relationships can be inferred. Go to the European Bioinformatics Institute's Clustal Omega program. This is the European Bioinformatics Institute's Clustal Omega program, which runs the Clustal Omega algorithm to perform a multiple sequence alignment. Leave the input type as protein, since we are running a protein sequence alignment. Then, open the file that you downloaded earlier, the FASTA file, and paste the entire FASTA file's contents into this box here. And scroll down and hit Submit. Once the program has completed, you can view the multiple sequence alignment in the Alignments tab, which is already open by default. You can also download the alignment file here, or you can view the phylogenetic tree. Some typical use cases would be to download the alignment file and view it in some sort of multiple sequence alignment viewer. However, we are going to be looking at the phylogenetic tree, so go ahead and click on the Phylogenetic Tree tab here. A phylogenetic tree is a diagram which shows the evolutionary relationships between species, organisms, or sequences. In this case, a multiple sequence alignment was performed, which compared the sequences to each other, and maps out their positions in the tree based on how similar they are to each other. Sequences closer to each other are more similar. Another way to perform the multiple sequence alignment in phylogenetic tree is to use Uniprot's built-in multiple sequence alignment tool. Go to Uniprot and at the top, select Align, which is here. One benefit to using Uniprot's built-in tool is if you are aligning protein sequences, you can use the Uniprot IDs of the proteins rather than the FASTA file. But since we already have the FASTA file sequences, we can just paste them in manually here. Then hit Run Align. Once the program has finished, hit the download button here. Make sure that it's compressed. And hit Go. Open the file and extract the contents. You now have a multiple sequence alignment file, denoted by the ALN extension, of the 5 sequences you aligned earlier. Keep it here because you will need it for the next step. Go back to the European Bioinformatics Institute website, but this time use their simple phylogeny tool. This is a simple phylogeny tool. You can drag the alignment file into the Choose File section. We're going to upload the multiple sequence alignment file directly. Note that we will be using the neighborhood joining method for clustering, which is sufficient for smaller sets of sequences. If you are running a larger multiple sequence alignment, it would be recommended to use UPGMA as the algorithm instead of neighborhood joining. Hit Submit at the bottom. Once the program is finished, you will find your phylogenetic tree. That concludes this video. You should now know how to compare your protein sequence to another similar protein sequence using Uniprot's built-in BLAST program, then generate a multiple sequence alignment file from the top few hits. Then, using the multiple sequence alignment file, know how to generate a phylogenetic tree to visualize the relationship between your group of proteins.